let's start with the embryological development of the brain. And so what I've done here is I have drawn out kind of loosely what you see in your lab manual right here. We have the primary vesicles right here, the secondary, and then these would be the mature brain regions. Here, these vesicles I should say, are present around the 4th week of gestation. That means mama has been pregnant for 4 weeks. She may or may not know that she's pregnant at this point. So it's kind of cool to see that our neural tube is already developing at this point. And then this is the 5th week of gestation. So very quickly you can see we start out as tubes and we kind of have these little pinches and things that kind of come off of that tube. And then by the 26th week of pregnancy, mama is about 6 months pregnant at this point. This is what these structures have developed into. So the way that I have kind of organized this is that I've done colors so that you can follow the colors all the way across, and then arrows. And what I want you guys to know is that, first of all, if I were to just point to any of these sections, could you just tell me what that is called? And then secondly, could you tell me how they are related? Meaning, do you know what primary vesicle the metencephalon came from? And how would you know that? How would you study that? You would just go back and say, okay, the metencephalon came from the rhombencephalon. We kind of remember these. Number one, there is a soft chalk drag and drop activity you can do to help you just get the answers. So you just drag and drop. That's not for grade or anything. So just drag and drop, get the answers, and then copy those in your lab manual, and then study that. Here within the primary vesicles, the way I like to remember these is that the prosencephalon, kind of reminds us of first, so it's the first part. Mes, that stands for mesencephalon. And I like to use shorthand, especially for this type of stuff. For me personally, I really struggle with just memorizing long names. You guys have heard me say that a bajillion times at this point. And so I will often use shorthand notes to help trigger the longer names because these shorter names are easier to remember, in my personal opinion. So MES, M-E-S, stands for mesencephalon, and the mesencephalon actually means midbrain. So kind of think mes, M, midbrain, it's right in the middle. Okay, so that's how I like to remember that. And then we have the rhombencephalon, and it's kind of this funny sounding name, funny shaped section of the primary vesicle. So if we were to move on, we would see that that prosencephalon is going to then turn into the telencephalon and the diencephalon. I think telencephalon, this kind of sort of looks like a telephone. So that's how I remember that one. And then the diencephalon, di meaning two, I think that's kind of the second one down. You'll notice here that the mesencephalon, did that name change at all? It did not change. And so in addition to that name not changing, is it still in the middle? Does our little memory trick work? And you would see, yeah, that mesencephalon is smack dab in the middle. So that's how I remember that one. And then the rhombencephalon is going to turn into the metencephalon and then the myelencephalon. And these three words here are the long, big ones. And so how do I remember those three? I've underlined these letters here because I'm showing you that they are in alphabetical order. So if that's just one of those little tiny things that helps you understand the order or help you differentiate the terms, you're all the better for that. So M-E-S, M-E-T, M-Y, that is in alphabetical order. And that kind of helps me remember those big, long M words. And then as we go along over here, again, these are the mature brain regions. And when we talk about the models, those are going to be the terms that we're mainly labeling on the brain models and then the sheep brain as well. So you can see that the telencephalon is going to turn into that cerebrum. The cerebrum is just this outer portion of the brain, like that. So that telencephalon is going to mold and form and turn into the cerebrum. And then the diencephalon, that will form what is referred to as the thalamus. We'll talk about that more in a moment. But the diencephalon is actually representative of a region right here. And the reason why I have these notes right here, epi and hypo 2, it's because we have an epithalamus, which is right here. In the video that I'm going to post right after this session, or excuse me, after my AMP2 session, I point these out. So we have the epithalamus, the thalamus, and then the hypothalamus. So that's what my little notes here refer to, is that technically the diencephalon is all of those, but you just have one blank in your lab manual, so that's just the thalamus. And then here, the mesencephalon is going to turn or become the corpora quadrigemina and the cerebral peduncles. Again, I will talk about those more when we go over the brain model. And then the metencephalon will form the cerebellum and the pons. The pons is that structure right there. And then the myelencephalon is the medulla or medulla oblongata right there. So that is the, those are the basics of the embryological development. Again, for testing purposes, make sure you know what those areas are if we were just to point at it, but then study where it came from and then study what it turns into. So kind of go back and forth like that. That will be very helpful. Okay, so then next what we have here, get my lab manual out. I know we only have a couple people on the live stream right now, which is fine, but anytime you have questions you want, you can type those in the chat box. And it's almost like a little bit nicer when there are less folks. You know, I'd love to have a ton of people, but when there are less folks, you have more time for individual questions, which is good. Okay, so looking at objective 2, we have the structures of the adult brain on human models and sheep specimen. I do have a video that goes over the sheep specimen that will also be included on your Canvas links. So you don't have to worry about trying to figure out what in the world is located on the sheep brains. So our first term here, we have again, the cerebrum. What did the cerebrum come from? The telencephalon, which really came from the prosencephalon. So if we were to follow this all the way back, we would say, okay, the cerebrum came from the prosencephalon, that first part of the primary vesicle. And it's broken up. The way I like to teach this is that we kind of have these geographical regions of the brain and some specific structures we can look at to help us organize these different parts. And what you have listed in your lab manual are lots of grooves, okay? And so we have deep grooves and shallow grooves. And this terminology is actually, should sound familiar at least, because we looked at them with the spinal cord. And whenever we have a deep groove, that deep groove is going to be called a fissure. A shallow groove is going to be called a sulcus. And if you see that word sulci or sulci, that's just plural for one singular sulcus. So when we're looking at the cerebrum, we can see it's very wrinkly. We have raised portions, and then we have grooved areas. And the grooves are going to be either a fissure or a sulcus. And then the raised portions are going to be the gyri, which is plural for singular gyrus. So let's start by looking at some of these grooves, and then we can talk about the lobes, because the grooves help create the borders for the lobes. So the first one we have here, so actually make sure we understand, which I think you guys can get this. This is posterior, anterior, and then this is the left side, right side. How do we know it's left and right? How do we know it's anterior, posterior? That cerebellum is on the posterior and inferior part of the brain. So if you can identify that, you know you're looking that this is the back side of the brain, I should say. So therefore, this is the front side or anterior side, and now we're just figuring out left and right. This longitudinal fissure is what creates the two cerebral hemispheres. Hemi means half, sphere is describing the basic shape of the brain. And so here, right here, this longitudinal fissure, think, okay, she said fissure. What is a fissure? A fissure is a deep groove, and it goes all the way down to these structures here. We'll go over this in another minute here, but these are the corpus callosum, but that's how far down it goes. So you can see it's super deep, and that's what creates the two cerebral hemispheres. Then we have the central sulcus. So the central sulcus is going to be this groove right here. Okay, remember, a sulcus is a shallow groove, and if you can see this right here, that central sulcus, so whereas the longitudinal fissure created the two cerebral hemispheres, what do you think, what two lobes do you think that central sulcus creates? Maybe you can take a moment just to think about that to yourselves. Okay, that would be the frontal lobe and the parietal lobe. So my right hand is on the parietal lobe, my left hand is on the frontal lobe. That central sulcus is what divides the two. Then, so there are two indentations, two specific gyri, and I do have a specific part in the video, and I'll just hold this up to show you. You'll see in the video I use this brain, and I have two gyri that are color-coded specifically. These two are representative of those indented gyri that you see on your list. So the line in between the two gyri, that's your central sulcus. The precentral sulcus, or excuse me, gyrus, is indented under the frontal lobe. So what do I say frequently in the semester? I say if a term is indented, it belongs to the term above it that is not indented. It's somehow related to it. So what we're saying is when you see that the precentral gyrus is indented under the frontal lobe, then that must mean that the precentral gyrus is located within the frontal lobe. And when we look at this model right here, we can see that this precentral gyrus is on that frontal lobe. Okay, this purple one right here, that is the post-central gyrus. Okay, take a moment to look at your lab manual. What term is that associated with, or what would you just say based on what you're seeing here? This is the parietal lobe. So that's why you see post-central gyrus associated with the parietal lobe there. These two raised portions, these two gyri, are important because they serve motor and sensory functions. So of all the raised portions here, they're all important. Why are we just focusing on these two? Because they innervate specific parts of the body, and they will either control, so the motor cortex, which is this red one, the motor cortex is the precentral gyrus. Also kind of think of that word precentral. If this is the central sulcus, and we're going in the order from anterior to posterior, think precentral kind of makes us think of before the central sulcus, and that's exactly what it means. So then post-central gyrus is going to come after the central sulcus. So the precentral gyrus is also known as the motor cortex, And that part of the cerebrum is going to control skeletal muscles. And in the video, you'll see me explain, and I'll show this picture of the cortical homunculus is what we call that. There is one for the motor cortex, meaning there is an image of a very kind of strange figure that is proportional to how much of the cortex controls that part of the body, the skeletal muscle in that part of the body. So it's a very weird, goofy image, but in the video, I actually put that on the screen so you can see that, and it is in your textbook. And I'm so sorry, I don't have the textbook right here with me. Otherwise, I'd give you the page number right now, but it is in your textbook. If you type in homunculus, you'll see what I'm talking about. So that pre-central gyrus is the motor cortex. The post-central gyrus is the sensory cortex. So kind of going back to what we talked about with motor function and sensory function, motor function is going to cause a movement. Sensory information is going to come from somewhere and then like come into us, if you want to think of it like that. And it's the same thing. We have a sensory homunculus, meaning we have another figure. It's very kind of weird looking, but it's proportionate to how much of the cortex receives sensory information from that part of the body. So again, in my video, I will show that image. And that's why we care specifically about those two gyri. The next one we have, okay, we have the lateral fissure. And the lateral, so before I already showed it to you, but when you hear lateral, you should be thinking about the stuff we learned our first couple of weeks together. Lateral, that's a directional term. So it would be a great starting point to go to the lateral part of the brain. And the lateral fissures are on each side of the brain. I have also seen these labeled as lateral sulci or lateral sulcus. So it really just depends on what term you see. But as long as use the term that we're using in the lab manual and you'll be good to go. But just FYI, it was actually several years ago, a few students brought that to my attention. They said, oh, I found in this resource that this is also called a sulcus. What will you accept? And of course, I personally would accept either because that's only fair. So just know that lateral fissure is going to create the border between the temporal and parietal lobe. So you can see there's our temporal lobe, parietal, lateral fissure. And then finally, we have those occipital lobes. Remember, we have right and left because we have two cerebral hemispheres. So those occipital lobes are simply where in our occipital region. So we don't need to overthink that one or anything. Okay, so that is that for the basic landmarks of the brain. Okay, let's see. If anyone, I'll check for questions. And if you don't have any, that's also fine. Just want to make sure I don't skip over anything. Okay, the ventral view. So for you guys, this ventral view here, what I want you to get from this are the cranial nerves. And I did make, if you are a subscriber, or even if you aren't a subscriber, but you have bookmarked my YouTube channel, you'll see that I actually uploaded several of these videos. The only one that is not uploaded yet is the video over this model that we're covering right now. But I do have a video called Cranial Nerves, and that addresses this figure right here. There are some terms on here that I just, I'm not sure exactly why they're listed here because I don't think they're great visual representations of what we have here. It's just because they're really hard to see. So for instance, okay, here we have These are the olfactory bulbs. Those we can see very clearly. Okay? Olfactory bulbs right there. These passageways then are the olfactory tracts. What is olfaction? That's our sense of smell. Okay? Those olfactory bulbs will lead to the olfactory tracts, or really it's kind of vice versa. And those we can see just fine and dandy on this image. The other ones that we can see just fine are those optic nerves, cranial nerve 2. And that's what these two are. They are cut off in the picture and on the model. Next week, we're going to talk about 2 of the sense organs. We'll talk about vision and hearing. And when we go over the eyes, we will see that these nerves are actually going to continue. They come out of the back of the eye. And so they're not, they're going to be longer than this. They're just cut off right here. That we can also see very clearly there. In the middle, okay, in the middle right here, see how this kind of looks like an X? Right there in the middle is a part called the optic chiasm. You'll see that word chiasm. You'll hear that quite a bit, and I talk about this in A&P2. Actually, this week, the same week in A&P2, we're going over the male reproductive system and basic genetics, and chiasmata comes up. And I say, hey guys, remember in A&P1 when we talked about the optic chiasm? So it would serve you well to really learn that term. But that would be right about here in the center of that X. And this is where we have crossing over. So what is a chiasmata? It's when we have crossing over of information, meaning, and maybe you've heard some of this before, some of the visual sensory information, let's just start with the right eye. Some of that information is going to come into my, visual information will come into my right eye. Some of that information will continue. You'll probably talk about these tracts in lecture. They'll go to the right cerebral hemisphere. But other pieces of information are going to travel down that optic nerve and then decuss or cross over, and that information is integrated by the left side of my brain. So some information that comes in the right side of my eye will go in, hit that optic chiasm, cross over, gets integrated here. Likewise, some information from my left eye will do the same thing. It will cross over and get integrated by the right side of my brain. So that is the importance of that optic chiasm right there. Let's see. So a term on here that's kind of strange, why it's on here is the hypothalamus. I'll show it to you on here. Again, I'm not in charge of making the quizzes, but I personally don't ever ask the hypothalamus on this view because we can see the hypothalamus better on this view, and you had better know what the hypothalamus, where the hypothalamus is on this view. But just for sake of going through our terms here, that hypothalamus is going to be this under region right here, okay? It's not this thing right here. That is the infundibulum that goes to the pituitary gland. It's not these things. Those are mammillary bodies. It's the part that would be basically behind on the page, those structures right there, or really deeper, okay, deeper into the brain, because if I were to show you, this area is the so right here, this is your hypothalamus. If I were to keep that pointer there, hopefully you're seeing how there's my hypothalamus. When I turn it like that, do you see it really well? My personal opinion is no, but just FYI, that's where you would label it in your lab manual, in case you are asked that. The other things that are listed here, which you can see, but there is, again, in my personal opinion, you better know it on this mid-sagittal view, and I will go over these with you, are the cerebral peduncles. the cerebral peduncles on this ventral view are these tiny little slivers right here. Okay. Yeah. Me personally, I think it's better to ask that question on this view, but again, I'm not making the test. So just be aware that on that view, that's where the cerebral peduncles are. I'm going to talk about those in a lot more detail on our next figure. Okay, then we have the oculomotor nerves. Okay, so let me put my brain back together. Okay, the oculomotor nerves are cranial nerve 3. And the easiest way to find those is this big structure here is called the pons. Okay, and that's what this is right here. And the oculomotor nerves are these two, let me put this up closer, are these two right here. Okay, those two yellow nerves that are coming out kind of the anterior side of the pons. So right here you can see, let me put my blunt probe on it before I turn it around. That's those right there are the oculomotor nerves. Those will help control the eye muscles, and we get to look at those eye muscles next week, which is pretty fun. In my video, I talk about all of the nerves, but obviously you see here that you only have some of those nerves listed. So just as is always the case, you know, what are you responsible for knowing? Only what's in the lab manual. But sometimes I cover more because, you know, I don't know, I just made the decision, if I'm going to make a video about cranial nerves, I'll just talk about all of them. I don't know how many of them you cover in lecture. So even if you aren't using all 12 cranial nerves for lab, I want you to be able to use my videos for lecture as best you can. So that's why you might hear me, or why you'll hear me talk about more than what is in your lab manual. Oh, I need to drink some coffee or something. Okay, so the next we have the trigeminal nerves, cranial nerve number 5, and that is going to be this portion right here. Think tri means 3. There are 3 divisions of that nerve, and that nerve actually is going to come through, and it kind of makes a pattern like this on the face. I remember I actually saw a couple times when I was in the ER, patients coming in with trigeminal neuralgia or pain sensations from that nerve, and it just looked terrible because that nerve will go to the facial muscles. Those patients I saw were very sensitive to any kind of pressure, even just like wisps of air kind of just triggered pain, and it just seemed awful. But when you think trigeminal, think of those 3 parts right there. And then I did show you the pons. So the pons is going to be this portion right here. Again, you can see it well on this view, but I'm going to point the pons out again on the mid-sagittal view as well. I think it's a little bit better representation of that. And then we have the medulla oblongata. That's this structure right here. Medulla oblongata is super important because that's where we have controls for heart rate, blood pressure, respiratory rate. So just showing you very quickly on this model here, this is where we have the pons, and then this is the medulla oblongata. And let's see, okay, the vagus nerve. So the vagus nerve in your lab manual, so go to the pons, and let me make sure you guys can see this well. So it's this one right here. Of the 2 that are here, it's the lower one. That is cranial nerve 10, or the vagus nerve. And the vagus nerve is a cool nerve. it is called the wandering nerve because it starts there in the brain and it innervates all sorts of things in the body. It's going to go to our pharyngeal muscles, laryngeal muscles. So even things like how we speak can be affected by the vagus nerve. It will innervate our heart, our lungs, liver. Actually, a conference I went to last year, they were talking about the latest research on the vagus nerve and how they're finding that it's even connected to the bladder. So it's just kind of cool. Why is that a big deal? It's because when you have something that connects to a lot of different things, if that thing gets damaged, well, we have some things to consider, right? So anyways, lots of cool research going on with the vagus nerve, and that is cranial nerve number 10. Okay. Oh, quick funny story. I normally share this in the live class. I'll share it to all 4 people watching this and anyone who's watching the recorded version of this. I will never forget one time in the ER. So normally when patients come through triage, they give the complaint, and so someone types in what the patient's complaining of so that we can kind of figure out who needs to be seen quickly. And this was what was written. Okay, do you guys see what is wrong with that? Vagus nerve pain. Okay, first of all, what is vagus nerve pain? Okay, or just saying, I don't even know. Second of all, is that how we spell Vegas, as in vagus nerve? -1. How do we spell Vegas? Oh my word. So anyways, guys, the spelling matter. Yes, I'm talking to an empty classroom right now, and I'm still preaching spelling, right? That's not a mistake that, changes the outcome of a patient or negatively affects a patient. But it's just one of those things that's just goofy as all get out. And a healthcare professional wrote that, okay? Healthcare people type those things. So I was just one of those things I'm like, oh my word. Okay, anyways, I digress. Okay, so for cranial nerves, watch my video, which is available on my channel right now. I just haven't uploaded it to Canvas because I won't put things on Canvas until I get that last video edited and uploaded as well. Okay, let's go to the mid-sagittal view. Mid-sagittal, you guys know that cut. Pull it apart. And now we're looking at it like so. So let me use this view, which matches your lab manual. Again, things are indented. We're telling you things are organized. So the first thing is the corpus callosum. And the corpus callosum is going to be this white body right here. Okay, so the corpus callosum is made of white matter. This is actually what connects the 2 cerebral hemispheres. So remember how earlier when we were just talking about that longitudinal fissure, I said it's a fissure, so it's very deep. It goes all the way down. Well, how do these 2 halves stay together? That corpus callosum. So that's what that structure is right there. Okay, then we have the diencephalon. So remember the diencephalon, I told you when we were looking at our embryological development, the diencephalon is right here. That's a secondary vesicle, okay, and that turned into the thalamus. But remember, I was like, it's really the epithalamus, thalamus, and hypothalamus as well, because all of these make up that diencephalon. So the thalamus is going to be this portion right here, okay? And this is hard to see in a 2D sense. So I hate that for you guys because there's nothing we can do about having just kind of like flat examples here, you know, in a video format. But just know that because we're looking at a half of the brain, the thalamus is an oval body that fits into each cerebral hemisphere. So it's kind of shaped like, let's see, maybe I can kind of, okay, this is terrible. I'm not even going to try that. You guys are like, oh my word, what is this lady doing? So right, left. What I've attempted to draw here is that the thalamus from an angle, okay, if this is the angle of the brain, okay, if this is the angle of the brain, like this, it's located inside, meaning this part goes in, it's inside that cerebral hemisphere. So we're only seeing the medial portion of that thalamus. Okay, so then the left hemisphere has its own part, and that's what would be coming out towards us. So I don't know, maybe I shouldn't even have drawn anything, but that is the thalamus. It's challenging to see on a 2D image like this. This little, I don't know, this looks like a little bird to me, like a little vulture baby or something. But that little, the eye of the bird, that is called the interthalamic adhesion. Not a term you have to know, but I'm just explaining to you that that part connects the two halves of the thalamus. So it's in between each cerebral hemisphere. Then we have the hypothalamus. Okay, the hypothalamus is going to be the beak of the bird. That's that part right there. And kind of think hypo, below, so below the thalamus. And then we have the pituitary gland and the pineal gland. So let me show the pineal gland first. The epithalamus, so remember here, I just had to know, again, you don't have to know epithalamus directly, but I kind of think it's helpful to know it. The epithalamus is this area up here. So kind of think epi above, it's up here. And the pineal gland, which is that structure right there, is a part of the epithalamus. Okay, so the pineal gland, which is responsible for creating and secreting melatonin, which is what helps regulate our circadian rhythms. That is a part of the epithalamus, and it is on the posterior side. So that little thing right there, that little doodad, is the pineal gland. And then we have the thalamus right here, and then the hypothalamus is right there. Then the pituitary gland is going to be that structure right there. Okay, that little stalk figure, that is the infundibulum. And this area right here, when you guys talk about hormone regulation and secretion, that is where all of that is happening. The hypothalamus is going to send signals via that infundibulum to the anterior or posterior pituitary. And then, yeah, man, lots and lots of stuff happens. Then we have the midbrain. Remember which term up here refers to midbrain. Okay, hopefully you're thinking, ooh, midbrain, middle, mesencephalon. So here, mesencephalon, what did we say we're going to see here? Corpora quadrigemina, cerebral peduncles. So it's kind of cool to tie that together. The cerebral peduncles are going to be this area right here. My blunt probe is pointing to them. So it's just this part these kind of like rolls or whatever. Those look so weird to me. Okay, those are the cerebral peduncles. And those are motor tracts. Okay, a tract is going to be composed of white matter. By the way, when you're looking at the brain, what do you notice? The spinal cord, what did we have on the outside? We had a lot of white matter on the outside and gray matter on the inside, those gray horns. The brain is the opposite. We have a lot of gray matter on the outside and white matter on the inside. And when you see that word tract, tract is describing bundles of white matter. So those cerebral peduncles right there, those serve as motor tracts. Okay, then we have the corpora quadrigemina. Okay, also I want to point out the midbrain. The midbrain is going to make up kind of this area that my finger is covering. You can see it's kind of in the middle. So it's a little visual and auditory cue for that. The corpora quadrigemina is going to be these two little bumps right there. See how we have an upper bump and then a lower bump? Quad, what does quad make you think of? 4, so 4 what? There are these structures called colliculi. You don't have to know them for me, but I like to explain them because it helps you actually learn what that is. Rather than just memorizing this complicated word, let's actually kind of understand a little bit about it. Here, because we have a mid-sagittal section, we're only seeing half of the group of 4. We have 2 superior colliculi and 2 inferior colliculi that are like this. And so that would be the view if you were to look at the back of someone's head, you would see these four projections kind of coming out. The 2 top and the 2 bottom. So the superior colliculi and the inferior colliculi. Well, what happens when we go to a view like this? You're only seeing 2 of them, right? So that's why you're seeing kind of an upper bump and a lower bump. Okay, those are visual and auditory. Those are responsible for visual and auditory sensations. So we've got visual sensations being processed here, auditory right there, which is pretty cool. And together, because we have 2 on the top, 2 on the bottom, that's where that word quad comes from, quadrigemina, the corpora quadrigemina. And then in the area of that cerebral aqueduct, okay, the cerebral aqueduct is actually a part of the cerebrospinal fluid. So that is this area right here, this little tunnel. Okay, so I'll show that to you on your lab. That's this little tunnel right there. Okay, right here we have the pons. Pons means bridge. So let's go back to these motor tracts up here called the cerebral peduncles. Peduncle actually means little foot. So the way I like to teach this is that the little feet are standing on the bridge. The little feet are standing on the bridge. The cerebral peduncles are on top of the pons. And then this area right here, this is our medulla oblongata right there. And then finally we have the cerebellum. Inside we have white matter called arbor vitae. Okay, arbor means tree-like, and it kind of looks like a pretty little tree. And then this space right here, that is the 4th ventricle. Again, the ventricle, when you hear me say ventricle, that's relating to CSF flow, which we're going to talk about next. So it's a lot easier to think of when we talk about it in that sense. Okay. Oh my word, I know that is a lot we're going over. So if you guys are watching this after the fact, maybe pause, go get a snack, and then come back because that was a lot we went over. So kind of give your brain a chance to digest this. For the sheep specimen. I have a video that's already available because I normally use that in class that goes through this. I want to remind you that there's nothing for you to label because it's already labeled. Woohoo, that's nice. But as nice as labeled pictures are, you might be looking at this and going, oh, what? All right, so it's good to watch the video. Normally, you would have done this. You would have been hands-on with the brains, which I hate that you're missing out on that experience now. But the video I have walks you through all of these terms. The other cool thing is on Canvas, especially for this week, we have lots of images that are blank and then labeled and color-coded. Again, those are great too, but sometimes even we kind of lose something in 2D images. So I think if you have the images pulled up or printed out, maybe watch the video so that you can see me like moving things around, and it might make a little bit more sense to you that way. Okay, so that is the sheep brain. So then objective 3, we have cerebrospinal fluid. Now the big thing about this, what I am putting in on Canvas is a screen recording to show you how to access some cool animations in your e-text. And I find that this part of the semester that you guys didn't even know you had these animations. And so there are some nice things that you can watch for any of the chapters actually, but this one's specific to this because I think it's really helpful for you. And I showed this video in class, but just click whenever I publish that, which will be later today. Just watch that screen recording. I'll show you exactly how to access the animations and then watch it. And that is going to bring this to life, which I think is super, super, super helpful. All right, so cerebrospinal fluid. We mentioned that in the last chapter with the spinal cord. This is where it comes from though. So in your lab manual, you have these little red squiggles. Okay, well, first of all, sorry, let me back up real quickly. Let me show you this model. And I do have a video. I did make a video. It's already published on my YouTube page. Again, I'm going to be putting that on Canvas later today. But I do have a video that just talks about cerebrospinal fluid. And so I want you to understand that this model right here represents spaces inside of our brain. If I have this is not to scale, by the way, but I think it does an okay job of showing this. This space is going to kind of snuggle in this area like so, but it goes into the brain. Okay, this part is going to go into that cerebral hemisphere, so that when we have both hemispheres like this, let's see if I can do this. Okay, imagine that I was able to smush the two halves together and close them. You would see that these two parts go into the cerebral hemispheres. And those are actually spaces inside the brain. And what is inside these spaces? Cerebrospinal fluid. And that's where it's created. There are, on this model, you can see these little pink squiggly things. That's what those pink squiggly things are in your lab manual. Okay, so you see how we have these like little pink squiggly things right here? Those represent the choroid plexuses. We have them in all 4 ventricles. Okay, so just like I'm showing you right here. And those are consist of, excuse me, these cells called ependymal cells, which help actually create the CSF. So that CSF gets secreted into these spaces, and then the fluid is just going to flow through all these ventricles and around the brain and the spinal cord. So we're really looking at how all of those fit together now. I recommend when you're studying this, go in order. These terms, CSF is flowing in all of these spaces all the time. But if we just want to kind of have some order when we're learning this, it's nice to have a kind of an arbitrary starting point. And so I recommend starting in the order we have listed here. You start in the lateral ventricles. So what are the lateral ventricles? That's what these are right here. This is just so I can give you a reference. Okay, so this is the anterior side. Okay, this is the posterior side. Hopefully you're seeing that well. And then this part down here, that is going to be inferior. And we'll see how all that fits together. So if we were to start arbitrarily in the lateral ventricles, we would say 1 and 2. Okay, steps 1 and 2. It doesn't matter which one is 1 and which one is 2 because they're both lateral ventricles. The only thing that really matters is that you understand, okay, because you're facing me, so this is how this would look in my head right now. This would be the left lateral ventricle, and this is the right lateral ventricle. From there, CSF is going to flow into the third ventricle right here. So kind of going back to that bird beak area. In your lab manual, it's that area right there in your lab manual. Okay. And then, all right, so third ventricle right there. From the third ventricle, then we're going to go into that cerebral aqueduct. Kind of looks like the neck of a bird. Okay, or mesencephalic aqueduct is how you'll see it on the sheep brain specimen. It's the same thing. And then this little pointy triangular part is the fourth ventricle. Remember, these are terms that we actually saw in that mid-sagittal view on the other page. That is that right there. So that's the fourth ventricle. So lateral ventricle, third ventricle, cerebral aqueduct, and then fourth ventricle is just this kind of triangular part that's associated with the cerebellum. From the fourth ventricle, we have these openings called apertures, which open up into the subarachnoid space. So let me just show you on your lab manual here. Okay, here we have these openings. This blue is representative of the subarachnoid space. We saw that term subarachnoid last week, didn't we? Subarachnoid beneath the arachnoid. We're referring to the meningeal layers. Okay. Remember we said that the meningeal layers cover the brain and spinal cord. We looked at the spinal cord last week, but I said that that's going to continue and cover the brain as well. So it's continuous. So the subarachnoid space up here, the fluid that goes here could also go into the subarachnoid space around the spinal cord, which is kind of cool to think about. So this blue part is the subarachnoid space. It will flow through there. Again, you can see how that's connected to the spinal cord down here. If we were to zoom in, these little projections, okay, this is actually projecting through the dura mater into an area called the dural venous sinus, or the superior sagittal sinus. I should say that one because that's in your lab manual. But if you hear dural venous sinus, I think I said that in my video, but I say both terms, but dural venous sinus is the same thing as the superior sagittal sinus. This is very descriptive, superior sagittal sinus. So this projection from the subarachnoid space into the superior sagittal sinus, this structure is called an arachnoid villus, or arachnoid villi just means many of these. Granulation, it's the same thing. You could say arachnoid granulation, or you could say arachnoid villus for one, or villi for many. Okay, so they mean the same thing. When CSF and the pressure inside the subarachnoid space is actually higher here, so that gets pushed out, and that's where we might have waste products, anything that needs to go into the venous blood flow will go into that superior sagittal sinus. So I will look at venous blood flow in AP2, but that is how CSF is related to blood flow, or how do waste products get out of the CSF via those arachnoid granulations into the superior sagittal sinus, and excuse me, that connects to the venous blood flow. Okay, so let's see. Last thing we have here are a couple of brain and cranial nerves associated with the cranial cavity. So very quickly, I want to point out a couple cool things here. Here's our from the skull. And maybe you're saying, no, I thought I could forget all that stuff. Oh no, we are constantly adding to our people puzzle. So we have to remember this stuff and specific things for this section. So when we're looking in here, the cristagalli. The cristagalli, remember, is part of the ethmoid bone right here. It's the part that kind of comes upwards. Okay, right there. What attaches to the cristagalli? It's going to be a specific part of the dura mater. So you could just know dura mater. That would be fine enough for me. We technically call it the cranial dural septa, because what happens is that dura mater, that outermost meningeal layer, is going to come around and then it goes down the longitudinal fissure. There are other places it goes into as well, because it helps anchor the brain into the cranium. But the part of that dura mater that comes down here is actually going to come down, and it will help anchor the brain to the ethmoid bone right there. Okay, so that cristagalli. Remember, why do we learn these bony projections and things? It's because most likely something has to attach to the bone right there. And that's exactly why we have a cristagalli on our cranial floor. So what's associated with that? The dura mater. If you want to get super technical, the cranial dural septa. Then we have the cribriform plate. Cribriform plate is right next to that. Remember that the cribriform plate had those little foramina, those little holes right there. So why do we have holes there? It's because those olfactory bulbs, you guys remember that from the beginning of our chat here? Olfactory bulbs, which are responsible for smelling, are going to rest in those depressions in the cribriform plate. That's kind of cool to think about. Okay, so when we, and this is not to scale, nothing I have fits perfectly together. That's just nice, right? Okay, so when I set this brain in here, if you imagine, you could see that olfactory bulb. When I set it in, it will kind of sit over that area where that cribriform plate is. But form follows function. Why do we have those little depressions there and those little holes? Oh, it's so our olfactory nerves can connect from our nose to our brain. Pretty cool. Putting our people puzzle together. Then we have the sella turcica. Remember that sella turcica is the Turkish saddle. The Turkish saddle is a part of the sphenoid bone. And so what sits in the Turkish saddle? The pituitary gland. So if I were to, let me turn this around. Again, this is not going to fit perfectly, but I'm just doing my best to show you that example. Okay, it's when the brain sits in there like so, that pituitary gland is going to snuggle up and be very well protected within that sella turcica. So pretty cool. And then last but not least, what is going to go through the foramen magnum? Remember, foramen is an opening, magnum is big, so big hole. What goes through there? The medulla oblongata. And really, the foramen magnum is the border where we have the medulla oblongata, the inferior portion of the brainstem, and then the beginning of the spinal cord. So the spinal cord is going to start here. Okay, so you could say medulla oblongata for that point, or beginning of the spinal cord, however you want to understand that, because it's kind of both. Okay, and oh my word, that is that. We have hit an hour. So for the folks that are here, let me know if you have any questions. But for those of you who are watching this after the fact, I know that is a lot, but hopefully me walking you through the lab manual and the images helps you get that filled out so you can study it and then do well in your quizzes. Just remember that I will be uploading the last video I have, which is talking over the brain structures, so all the stuff that we looked at over the mid-sagittal view. That will be uploaded in a few hours after I do my AP2 session. And as always, just let me know if you have any questions on Canvas. So I hope you guys are doing well. Let me know how I can help you. And I hope you have a great, restful weekend. Okay, I'll see you guys later.